This week we'll start learning the nervous system together. If you have reached this point in the course, then you've already passed bones and muscular system, which I think are two of the more challenging systems to learn. As we go through the nervous system, we'll find that we'll break it up into kind of manageable sections, but there's quite a bit of information to learn in this section as well. We'll have two primary divisions that we'll look at, the central nervous system and the peripheral nervous system. And then even within each of those, we will break it up into further sections so that you can learn the different parts of each section kind of in a manageable group of new names. We will start with the central nervous system and then we'll go to the peripheral. So within the central nervous system, there's the brain and the spinal cord, and we'll learn it that way. We'll learn the brain and then we'll learn the spinal cord separately. There'll be a lot more information in the brain to learn than there will be in the spinal cord. Then we'll move on to the peripheral nervous system, and we'll break that down into kind of regions of the body like we've done with bones and muscles. So you'll learn peripheral nerves of the upper extremity and then the lower extremity and so on. But in general, when we talk about the peripheral nervous system, we can talk about those nerves that are carrying information from the environment back to the brain. That would be the afferent, that would be the sensory neurons. And then we can talk about neurons that are carrying information from the brain out to, let's say, the muscles of the body in order to help you move your arms and legs and so forth. And we call that the efferent portion of the peripheral nervous system. And so you'll hear me use those terms. I'd suggest that you kind of get them straight here early on so that when I use them in the future, you'll understand when I say efferent, that means coming from the brain or spinal cord and going out to the body. When I say afferent, that means coming from the periphery, usually some kind of sensation, and going towards the brain. So afferent is bringing information to the brain, and efferent is carrying information away from the brain. We also have some other terms that we'll use talking about the nervous system, and we can split these up into sensory or afferent processes and efferent processes or motor processes. So if we look at the sensory side, we can break up the sensory information into two main groups. One is we call somatic. If you remember previously, we talked about how that word soma comes from the Greek word for body. And so somatic means basically, it's pretty much what you think of when you think of feeling. When you think of sensation, you're usually thinking of somatic sensation. You're thinking of what my fingers feel, what my head feels, what my eyes, or not my eyes, but what my muscles feel. For example, if you have achy muscles at the end of working out, what my joints feel. That's somatic. That's coming from the parts of our body that typically we think of. Visceral, we've learned that term previously, refers to the organs. And so we don't often talk about, how does my liver feel? How does how do my intestines feel today? So that they get kind of a separate category for sensation because they have a different entrance into the nervous system. Although, as we'll see, sometimes your brain is unable to tell the difference between somatic and visceral sensation. And so, for example, when your gallbladder hurts, that's a visceral sensation, but you may experience that pain as right shoulder pain, which would be a somatic sensation, because sometimes your brain has difficulty interpreting the signals that it's getting. And we'll talk about that as we go along in the course. But for now, just remember the term somatic refers to basically external stuff, skin, muscles, joints, and visceral is going to refer largely to organs. Smooth muscle as well, because the smooth muscle often surround or help comprise some of the organs of our body. So somatic and visceral. When we get to the motor side, we will continue to talk about somatics for the voluntary stuff. So largely Again, the motor system, what we typically think of is muscles contracting. I'm moving my arm, I'm moving my leg, I'm moving my fingers on the piano, I'm moving my mouth and my tongue around in speech. So that's skeletal muscle, that's voluntary muscle. We're going to call that somatic. There are some involuntary movements like reflexes that are also under the somatic nervous system, but it's because they affect skeletal muscles. So if we're talking about the movement of skeletal muscle, that's going to be somatic nervous system. When we talk about controlling smooth muscle or controlling organ muscle, heart muscle, or even glands, we're going to switch to what we call the autonomic nervous system. So instead of calling it the visceral nervous system, we call it the autonomic nervous system. So we'll spend some time talking specifically about the autonomic nervous system because it's quite important in the body and has quite a bit of information of its own. So we will kind of break that up into a separate section. Just to give you an idea of the flow of what we're talking about, over here we have receptors. This would be the afferent side. So let's just take an example. Let's say you reach out and you touch a hot pan on the stove. So you're going to feel that heat. That's going to be a peripheral receptor, right? It's going to be a somatic sensation. It's going to be something you felt in your skin, and it's going to send that afferent information up to the central nervous system. So you're going to end up getting to the spinal cord, and you may have a reflex going right back to efferent from the spinal cord, or it may also send some information up to the brain. But both of those are central nervous system, right? Brain and spinal cord. So the information eventually makes it from the periphery to the central nervous system. Once it's in the central nervous system, you get some kind of a processing of that information. Is this light touch? Is this a good kind of sensation, or is this a danger kind of sensation? In the case of a hot pan, that's going to signal a protective response. And so your brain and spinal cord will process that information, and it will then send out efferent commands, right, motor commands for muscles to contract, because once you touch that hot pan, you want to remove your hand from the pan, and that requires contraction of the muscles of the arm, maybe the muscles of the hand and wrist. And so that information is going to come from the central nervous system, from the brain, from the spinal cord, and it's going to send a signal to make those muscles contract so that you move your hand away from the pan. So that would be down the somatic nervous system, because we're talking about moving voluntary muscle, right? You might also get at the same time some kind of an autonomic response. You might get an elevation of heart rate, for example, as adrenaline kind of is released into your blood supply in order to get you ready for fight or flight due to the pain that you felt. That would be signals going down the autonomic nervous system. And you may get signals going down both at the same time, all starting from the central nervous system, the brain and or the spinal cord. So that's kind of the flow. Afferent is going to receive information, send it to the central nervous system. The central nervous system will process the information and then create a response. And that response is typically, if we're talking about the voluntary side, the somatic nervous system side, it's going to be contraction of muscle. It may also signal the autonomic nervous system, which is kind of involuntary and often involves smooth muscle, organ muscle, cardiac muscle, or glands. Okay, now that we've kind of got some general ideas understood, we're going to move on to looking at the anatomy of the nervous system. We'll start by looking at the anatomy of a neuron. And we'll start kind of with general anatomy, and then we'll kind of get a little bit more deep into the structures that are in the different parts here. And hopefully you've seen something like this at some point already in your life, but we've got 4 basic components to a neuron. We have the cell body, which is, the kind of looks like the cell part. It kind of looks like any other cell if you just took out the cell body. It's got a nucleus in it and some other organelles and cytoplasm and all that. But often attached to the nucleus, or sometimes coming off one end of the cell body, we find these things called dendrites. Now, dendrites are the portion of a neuron that receive information from surrounding cells, often from surrounding neurons. So the dendrites are these kind of spiny things coming off, in this picture, coming off the cell body. And they're going to receive information from other cells. Okay, on the other side of the cell body, we have the axon. The axon is the part of the neuron that carries an electrical signal that begins here at a place called the axon hillock, and that electrical signal goes to the end of the neuron until it is causes the release of chemicals called neurotransmitters here at the axon terminals. So the axon hillock is the area in between the cell body and the axon. You will find the axon hillock will be in between those two parts. That's kind of where they come together. And we'll see in physiology that that's the area of the neuron where the decision is made of whether or not to send an electrical signal down the axon. Now, once that electrical signal is produced, it's all or nothing. It's gonna go, and it's gonna reach the end of the axon to these points called axon terminals, and those axon terminals will then release chemicals called neurotransmitters onto their effector cells. So often that's another neuron, for example. So it's one neuron communicating with another neuron. So you'll say, this is where communication happens. And I would say the dendrites are also where communication happens on the other end, right? So if we could imagine two similar cells, the neurotransmitters released here at the axon terminal would then be received by the dendrites of the next neuron in line. And you see how the cycle would then repeat. So we have dendrites receiving information in tune with the environment around it and receiving signals from the external environment. Then we have the cell body, which is the bulk of the cell, just like most cells in the body. Then we'll have the axon hillock, which is kind of the connection between the cell body and the axon. Then we have the axon, which carries the electrical signal to the end, and the ends are called axon terminals. So that's kind of the general layout of a neuron. We'll see several examples, some which look like that picture, some which look a little bit different, and we'll talk about those. But so that's the general idea of a neuron, but there are actually different types of neurons and neural tissue, some of which are not even neurons, but are still involved maybe in helping the health of neurons, for example. So we have some what we call supportive cells of the central nervous system, and these are not the ones that are carrying the electrical signals or doing the communication between the brain and the muscles, but these are still important cells which make up part of the neural tissue, either in the brain, spinal cord, or even in the peripheral nervous system. And they all have special jobs to do, supportive jobs to do to kind of keep our neural tissue healthy. The the largest and most numerous of these supportive cells in the central nervous system are the astrocytes. So we're going to break up supportive cells into which ones are in the central nervous system and which ones are in the peripheral nervous system. So if we look at the central nervous system first, we'll look at astrocytes to start with, and astrocytes are the most numerous of the supportive cells. They also happen to be the largest. And they have several functions, in fact. You can kind of see a picture there. So here would be, there's a neuron and there's a neuron, and surrounding them, in between them, are multiple cells, one of which could be an astrocyte. So the astrocyte does several things. You can see that it regulates the composition of the fluid in between the cells, the interstitial fluid. We'll talk here in a minute about the blood-brain barrier, but that's created by astrocytes as well. They also help keep the neural tissue together, give some structure, some form to the neural tissue, because they have microfilaments that they can use to almost act as a skeleton for neural tissue. They're involved in repairing and scarring of injured tissue. This is very important. This is part of the work that Dr. Vora does, looking at repair and damage to neurons, and so astrocytes are a big part of that. And they also direct the growth of new neurons. So once neurons are injured, there may be some growth of surrounding neurons to take over the space that was controlled by the dead ones, or during development, during fetal development, and as an infant and as a young child, your nervous system continues to develop and grow, and often that's under the direction of the astrocytes. So you can see they're very important cells. They're very numerous, and they have several functions. So one of their functions I mentioned was the blood-brain barrier. And what we mean by that is the fact that the blood that flows through the brain is not exactly the same as the blood that flows through the arteries and veins of the rest of the body. And the reason for that is that whatever enters your circulation, so let's say you take a new medicine, and that goes into your stomach and then into your intestines, and it gets absorbed into your blood supply. And then that medication is all over your body. Once it gets into your arteries, it's going to go everywhere. Well, when it reaches the brain, there's actually a selective process that happens where the brain circulation will not allow everything to come into the brain. It will be very selective about what it allows to pass from the arteries coming to the brain into the tissue surrounding the brain. And we call that selection the blood-brain barrier, meaning there's a selective barrier between the blood flow and the brain tissue, where the brain can be protected from things in the blood that might be harmful. And that barrier is actually caused by the astrocytes. So what happens if we take a look at a normal capillary, and we'll see this more when we get into the cardiovascular system, which will be the next unit we do. But the capillaries are very thin-walled blood vessels that are composed of squamous epithelia called endothelium. And the way that capillaries work, the way that material moves in and out of capillaries, is because there are pores in between the endothelial cells that allow material to escape. That's water, that's electrolytes, that's other molecules like gases, it may be nutrients, it may be drugs that we take. They're able to pass in and out of the capillary because of the pores. Well, what happens in the brain is that those pores are actually removed. And that happens because the astrocytes that are surrounding those blood vessels in the brain secrete a chemical that changes the junction between endothelial cells. So whereas in the rest of the body, there's kind of a gap between the endothelial cells so that material can flow back and forth. In the brain, because of the chemicals produced by astrocytes, those gap junctions are taken away, and you get something more like a tight junction in between endothelial cells. So now you cannot get kind of free-flowing material into or out of those endothelial cells. So all of the material that's trapped inside the capillary is not able to get out and bathe in the fluid surrounding the brain tissue. Now, of course, we need some of that material. We need glucose, we need sodium and potassium, and so some of the small ones are able to, dissolve right across the endothelial cell, things like gases, for example. But things like glucose, they will need a special transporter. So the endothelial cells inside the brain will have special transporters that move only the material that is helpful across that blood vessel and into the brain tissue. So this is kind of a protective mechanism that the brain uses so that not everything from the blood makes its way to the brain. Now this becomes really interesting when you talk about clinical applications and you talk about giving medications. So some medicines either are small enough that they can diffuse across the endothelial cell, and so those medicines will make it into the brain tissue and cause effects there on the brain. Other ones will not be able to pass through. So I'll give you two examples. On the one side, You might have a medication that crosses the blood-brain barrier and causes side effects in the brain that you don't want. So an example would be like Benadryl. If you've ever taken Benadryl, for most people it makes you quite sleepy. And that's because of the effect that Benadryl has on the brain. Benadryl is such a small molecule, it's able to get past the blood-brain barrier and get into the brain tissue and make you sleepy. Now, most of the time, we're not taking Benadryl to get sleepy. We're taking Benadryl to help with our allergies, maybe to help with a runny nose or an itchy nose or a rash from an allergy. And because it gets to the skin, for example, and helps the rash. It gets to the nose and the lungs and helps with the sneezing and the mucus production. But it also gets into the brain, and what it causes there is sleepiness. Now, some people use that side effect as a sleeping aid, and if you look at Tylenol PM and medicines like that, it's basically Tylenol mixed with Benadryl. So they use the side effect to help people sleep. But what we discovered some years ago is that, I wonder if we can take the antihistamine medicine in something like Benadryl, and I wonder if we can make the molecule larger, add some more molecules to something like Benadryl, and make it too big to cross the blood-brain barrier. So that way we would still be able to get the effects of the drug in the skin, for example. It would still help rashes. We could get the effect of the drug in the nose, and it would help with sneezing and runny nose. But we wouldn't get the side effect of sleepiness because the drug would now be too large to pass the blood-brain barrier. And that's when we develop medicines like Claritin, Allegra, Zyrtec. These medicines all come from something like Benadryl, but they've been made too large to pass through the blood-brain barrier. That's why they don't cause sleepiness like Benadryl does. So we kind of use that information to our benefit. Now, on the other side, sometimes if you don't understand the blood-brain barrier, you might give a medicine thinking it's going to help the brain, but if it can't get from the blood to the brain because of the blood-brain barrier, then you're actually not doing any good. And so I'll give you an example. People who have Parkinson's disease, they have low dopamine levels. So we want to give them dopamine to help their symptoms. The problem is if you make synthetic dopamine and you ingest it and it gets absorbed into your blood, it does not pass the blood-brain barrier because of some of its properties. So researchers found a way to add a molecule to dopamine, and we call it levodopa. And that adding that molecule changed the shape and charge of that form of dopamine so that it could now pass through the blood-brain barrier. So now we're able to give patients with Parkinson's a modified form of dopamine that's able to cross the blood-brain barrier, get into the brain tissue, and be useful. Okay, some other supportive cells of the CNS, the central nervous system. Oligodendrocytes are important. They also have a kind of and that would be this looking cell here. They have some supportive actions as well. They kind of connect neighboring axons together and kind of hold axons in groupings. But probably more importantly, they produce myelin. And myelin is kind of a lipid-based substance that surrounds a lot of our nerves and helps the electrical signals to be transferred much more rapidly down that axon. And we'll see why here in a picture in a minute. So in the central nervous system, it's oligodendrocytes that produce myelin. Also in the central nervous system, we have microglia, and these are the immune cells. So there's kind of a picture there. We talked about how we have immune cells in the skin, for example, and immune cells in different parts of the body. These are the immune cells of the central nervous system. And they kind of eat. That's what that, remember, phagocytes are cells that eat other cells. And so they're going to eat viruses, bacteria, dead tissue, and kind of help keep the central nervous system clean of debris and hopefully help with the immune response. All right, I think this is the last one in the central nervous system, ependymal cells. These are an epithelial type cell that help produce the cerebrospinal fluid. So if you remember when we were talking about the surface anatomy, and I told you about meningitis and how you put that needle in between L3 and L4, or L2 and L3, and you can get a sample of cerebrospinal fluid and see if it's infected. Well, that fluid has to be made somewhere, and the cells that make that fluid that the brain and the spinal cord float in, those are called ependymal cells. All right, we're going to move to the peripheral nervous system and just mention a couple of cells here. Satellite cells, you can see well in this picture. They're going to be in the nervous system, the cells that help regulate nutrient exchange, kind of like the astrocytes do in the central nervous system. And you can just see them well here in a picture. These are like cell bodies of peripheral neurons, but you can see they're surrounded by all these darker looking cells, and those are the satellite cells that kind of surround and help regulate what's going on around those neurons in the peripheral nervous system. Also in the peripheral nervous system, just like we had oligodendrocytes in the central nervous system producing myelin, in the peripheral nervous system, the cells that produce myelin are called Schwann cells. And you can see here in the picture, like if the blue thing is the axon, and you can see the nucleus here of the Schwann cell. That Schwann cell is wrapping itself around several times the axon. And remember that the cell membrane of cells is largely composed of lipids, right? It's a phospholipid membrane. And so what that does is basically put a layer of fat insulation around the axon. And we're going to see here in a minute how that helps messages get transferred faster down an axon that's been myelinated. So here's a picture of what a myelinated axon looks like in the peripheral nervous system. And you can see that there are multiple Schwann cells surrounding themselves around the axon of this neuron. So for example, there's one Schwann cell, there's another one, there's another one, there's another one. And each one is wrapping itself around a portion of axon, kind of insulating that axon by wrapping it in this fatty membrane. And what you'll notice is that there are areas in between the myelin, there are areas in between the Schwann cells which are not wrapped in this fatty material, right? So this area here, for example, this area here, you can see the areas in between the myelin. And the areas in between the myelin are called nodes. And they were named after a guy named Ranvier, so we call them the nodes of Ranvier. And those become important in physiology. So right now I just want you to learn the idea of axons being wrapped by Schwann cells forming myelin sheaths, and in between the myelin sheaths are the nodes of Ranvier. We sometimes call the area in between the nodes, the area that's actually wrapped in myelin, we call that myelinated internodes. They're internodes because they're in between the nodes. And then you can see here's the cell body, there's the axon hillock, and there's the beginning of the axon. Then you've got the myelin wrapping it all the way to the end. So here's just a picture. We'll talk about this more in physiology, so don't worry too much about the how this works. I just want you to understand that it does work, and what's the role of the Schwann cell and the myelin. So let's just so that you can understand it in context, let's say we have a cell body and it's been triggered by another axon, other neurons around it that it needs to fire a signal, right? So it's going to reach the axon hillock. Sorry, I lost my pen here. It's going to reach the axon hillock, and it's going to trigger an electrical response. And so that electricity is going to move down the axon, okay? But in a myelinated cell, it's going to do that in a jumping fashion, okay? It's going to move from node, and then there is no way for electricity to enter or leave the axon where the myelin is, so it's going to jump. And by jumping, by that electrical signal jumping from node to node, that allows the signal to be carried down the axon much faster. We have some very long axons in our body. For example, the one from your pinky to your spinal cord, that's really long. And if we had to rely on kind of traditional movement of ions down that axon to produce an electrical signal, it would take a long time. and you would probably do damage to your fingers, for example, before you ever got the signal to remove them from the heat. So we need to speed up the process of that electricity moving and causing signals, and we do that by wrapping the neuron in myelin, and that causes this jumping kind of electrical signal, so that electricity only occurs at the nodes of RON-VA. And again, we'll talk about that more in physiology. So right now, just understand that it's produced by Schwann cells in the peripheral nervous system, oligodendrocytes in the central nervous system. It helps to speed up, and the electrical signal happens in those nodes of RON-VA. All right, and that kind of brings me to a discussion of white matter versus gray matter, and you've probably heard those terms. White matter, the whiteness of the white matter, the light color of white matter, comes from myelin. It's that fatty sheath is what leads to the lighter, more white color. And they are axons, right? So myelin only occurs in axons. So when we talk about white matter, we're not talking about cell bodies. We're talking about axons. So wherever you see white matter, that's just all axons, really very limited or no cell bodies there. And they're not just axons, but they're myelinated axons, and that's what gives them their color. Gray matter, on the other hand, is a darker color if you look at it under a microscope or you look at it even in real life. And the gray matter is composed largely of cell bodies, or of unmyelinated axons. So because they don't have the myelin, they don't have the lighter color, they look a little bit darker, and we call that gray matter. And so, for example, you can see in the picture over here, this is a cross-section of spinal cord tissue, and the white matter is all the lighter colored stuff you can see, and the gray matter is this butterfly-shaped portion in the center. So the butterfly shape would have the actual neurons, and the white surrounding area would be the axons going up and down the spinal cord. So if we kind of go deeper now into neuron anatomy and we take a look at the cell body in particular, once we look at the cell body, we can see what's inside the cell body. And really, it's the same stuff that's in the rest of cells that we've already talked about, but we have a couple of unique names here that we have to learn because it's a neuron and not, a skin cell or something else. So, for example, the cytoplasm of a cell body of a neuron is called the perikaryon. So we have a new name for cytoplasm. And it contains pretty much what cytoplasm and other cells contain. It's got cytoskeleton fibers, it's got mitochondria, it's got, endoplasmic reticulum and Golgi bodies and ribosomes and all that, just like the other cells do. But one particular unique feature in a neuron cell body is what we call Nissl bodies. And these are important largely because it's what gives gray matter its gray color. So we were just talking about white matter and gray matter. The reason that gray matter looks darker, looks gray, is because of these Nissl bodies. So these are dark staining areas within the cell body, and they end up staining darker because of the ribosomes that are on endoplasmic reticulum. And so if we take a look here, so this kind of dark purple is a cell body, and if you zoom in, you can see these kind of individual dark staining areas right all over the cytoplasm. And those are the Nissl bodies, and it's basically what gives gray matter its gray color. If we move down from the cell body down into the axon, remember the axon hillock is that area right in between the cell body and the axon, kind of connects the two. The cytoplasm within the axon is a little bit unique in that it doesn't carry many of the organelles that the cell body does. And so we give it a different name. We don't call it the perikaryon, we call it the axoplasm. Should be fairly easy to remember. It does contain some proteins and structures within it, like cytoskeleton tubules. So in order to kind of keep its structure intact, it does have some cytoskeleton tubules. There are some enzymes and mitochondria needed to help move material from the cell body to the axon terminal. But there really is no production happening in the axon. There are no ribosomes, no endoplasmic reticulum, Golgi bodies, all these things needed to make proteins. The axon is really a place for transport, and so it has really only what it needs for transport. If we get to the end of the axon, we have what we call telodendria. These are kind of some branching that may occur at the end of an axon, and that just kind of helps that axon have an effect on different parts of a surrounding cell or on multiple cells by kind of breaking up into multiple tentacles almost, telodendria. And then these telodendria will end, the axon terminal will end, you can see there, they look like little feet. And we can call them axon terminals, and that's fine, but you'll also see the term bouton here, almost like a little button down there. So axon terminals or boutons. Okay, so we've been looking kind of at a representative neuron, and we've been making it look a certain way, but just so you don't get the idea that all neurons kind of look like that, I wanted to show you what other neurons may look like. And there are 4 structural groups of axons. So cell bodies are cell bodies, and they pretty much look the same. What makes one neuron look different from another is what's going on with the dendrites, with the axon. And so we'll look at 4 groups here. So the first is anaxonic. These are small neurons, and an, right, means without. So anaxonic means without axons. So it's just a cell body with a bunch of dendrites, and there's really no axon here. Now, it's not that there are no axons, it's just that you can't, from looking at the cell, you can't determine where the axons are. They all look like dendrites. So some of them are receiving information and some of them are relaying information. It's just from looking at it, you don't know which is which. They all look like dendrites. And as far as function, we don't really understand them well. We just know that they exist. We'll see on some of the other ones, we understand them a little bit better. So another type of neuron would be a bipolar neuron. And that's going to have the cell body in the middle. And it's going to have one long dendrite with lots of branching coming off on one end, and then one long axon with multiple telodendria and terminals coming off the other side. And the cell body is right in the middle, so you get the dendrite coming off one side of the cell body, and you get the axon coming off the opposite side of the cell body. These are unmyelinated, and these are what we see in some of our sensory neurons. A pseudo unipolar neuron looks very similar to a bipolar, except that the cell body is kind of off to the side. So you have one long axon, and one end that branches off into dendrites, and the other end it branches off into telodendria and axon terminals. And the cell body is still in between the two, but instead of the dendrite coming off one end and the axon coming off the other end, it's one long axon with 2 ends, and the cell body is kind of off to the side. And we can see these are also some of our sensory neurons, but these ones are often myelinated, so they're going to have a faster signal. This is the one that we've been looking at. This is kind of the representative neuron that we always show in pictures, and that's partly because it's the most common. There are lots of neurons that have multiple functions that look like this. Motor neurons, for example, are multipolar neurons. And multipolar just because you have multiple dendrites coming off of the cell body. So that means that neuron can receive information from multiple axons from multiple neurons that are surrounding it. Then the cell body will process that information and decide whether or not to send a signal down the axon and into the terminals. So that's kind of an anatomic way of classifying neurons. Are they anaxonic? Are they bipolar? Are they pseudo-unipolar? Are they multipolar? But we can also classify neurons in terms of their function. Are they sensory neurons? That means they're afferent, right? Afferent means they're going to carry information to the central nervous system. So are they sensory neurons? Are they motor or efferent fibers, meaning they carry information away from the central nervous system? Or there's a third classification called interneurons. That means they don't actually carry information to or from the central nervous system. They actually occur in between cells within the same system. So in between two peripheral neurons or in between two central neurons, you may have a neuron that just connects two of those, two afferent neurons or two efferent neurons. And it's just kind of a relay station. They do have some function, though, where they can kind of modify the signal that's occurring between the two neurons. So those would be interneurons. So just to give us kind of some example, put it into context here, we can see what this might look like. So let's say we have a receptor. Let's go with, how about position sense? So inside your joints, you have receptors that can tell if your finger is flexed or your finger is extended, and they send that information to the brain so that your brain knows where your finger is at all time. So that information is sensory information, and you're going to send that through afferent fibers, right? And it's going to eventually make its way to the central nervous system. So it's going to do that first at the spinal cord. Well, it may be that, let's follow this blue line down here. So we're going to enter the spinal cord. Now, you might have a reflex that actually bypasses the brain altogether and just immediately causes the movement of your hand because maybe it was a dangerous signal. So you went from afferent to central nervous system to efferent directly. But often what happens is you're going to go through the central nervous system, and that neuron is going to synapse with another neuron, and that second neuron is going to be the one that then synapses with the one that's going to make the decision. Then that neuron makes the decision, and you get an efferent signal sent out. That could also be done kind of involuntary through the autonomic nervous system, and you would get the same kind of a flow. So afferent, maybe some interneurons to efferent, and then you get a response. So let's look at what happens down at that axon terminal. So we talked about the cell body and we talked about the axon. Now we're going to move into one of those terminals and look at what happens there that allows communication to occur from one neuron to its target tissue. And that target tissue, again, might be another neuron, or like in this example, it might be a muscle, or it could even be a gland. So let's look at what happens. You get that electrical signal comes down the axon, and when it reaches the terminal, it's going to cause the release of these chemicals, which are shown by the green circles. These are neurotransmitters, right? So we call that electrical signal that comes down the axon, we call that the action potential. And that's going to be super important in physiology, and we'll spend quite a bit of time talking about that. When that electrical change caused by the action potential reaches the terminal, it causes the release of the neurotransmitters. Now, the neurotransmitters have to cross this very small area, right, in between the terminal and the target tissue. And that space that the neurotransmitter is dropped into is called the synaptic cleft. And then once that neurotransmitter crosses the cleft and lands on the target tissue, it will then cause a response in the target tissue. If it's another neuron, it might just cause another action potential to be generated. If it's a muscle, it might lead to contraction of muscle. If it's a gland, it might lead to the secretion of a hormone or something like that. And here's just a reminder that action potentials or neurons can have an effect on multiple types of cells, other neurons, muscles, or glands. I just threw this picture in there to give you an idea. You know, we talk about neurons in isolation often. We talk about one neuron communicating with one other neuron. But the truth is, in your brain, in your spinal cord, there's often many neurons that are all communicating with one neuron, and then that one neuron may then communicate with multiple neurons. And so just to give you a picture of how complicated it actually can be, look at all the blue ones are axon terminals from other neurons that are all synapsing on this one cell body, which is the purple underneath all of the blue. And so the cell body then has to kind of integrate all that information. Some of those cells are saying, produce an action potential, and some of those cells are saying don't produce an action potential. And so it has to kind of add all that up and decide whether or not it's going to send a signal down its own axon. And so it's a bit more complicated than we show here. So I just wanted to kind of give you a picture that's a little bit more realistic. So on the same token, so that's kind of receiving information, but on the same token, one neuron may communicate with multiple other cells, whether it's multiple muscle fibers or whether it's multiple neurons. One neuron may communicate with multiple neurons on the other end. So if the signal begins in one neuron and then kind of spreads out from there, and you get a kind of a multiplication of communication, so the one neuron talks to two, and those two each talk to three, we're going to call that a divergent pattern or divergence. So that's one neuron is being spread to many. And this, for example, is like visual information that's sent to the back of the brain and to balance centers at the same time. So you get one photon of light that hits the retina, but then that signal is sent not only to the back of the brain to give you conscious sight, which maybe this would be conscious sight on this side, but the information is split and also goes to the balance centers of the brain to help you keep your body upright, for example, when you're running. So you get a divergence of information. It started with one neuron that received the light, but then it sent that information off to multiple areas of the brain. You might get convergence. That means you get information coming from multiple neurons all converging onto one neuron. That was kind of like the picture I showed you a couple of slides ago. And one example there might be respiratory centers. So you might get, for example, the autonomic nervous system is sending a signal to your diaphragm, to contract every 2 seconds, every 3 seconds so that you breathe. But you might be able to send another signal to hold your breath, right? So you can have the involuntary signal coming and trying to make your diaphragm contract so that you breathe, but you can override that signal consciously by holding your breath. And so you kind of mix those signals together. You have more than one signal going to the same muscle at the same time. We would call that convergence. You can have serial processing. That means one neuron tells one neuron tells one neuron, and there's no convergence and no divergence. And this would be like pain sensation in the brain. You get one receptor on your index finger, for example, receives pressure, and it's going to send that signal to one particular part of the brain so that your brain always knows when that part of its brain is stimulated, it means that part of the finger was touched. And so that's what we would call serial processing. It goes one to one to one. And that's important for organization within the brain, for the brain to localize information on the body. The serial processing is important. You might also have parallel processing, where the initial signal is broken up into multiple. So you have kind of initial divergence, but then you have serial processing after the initial divergence. And this is an example of this might be pain, for example. So the brain still needs to know where the pain occurred. So you still need serial processing along one line so that it ends up landing on the part of the brain that tells you where the pain is. But you might also need, several other pathways may be initiated. For example, you might get a pathway that goes to your voice and you might say, ouch. You might get a reflex pathway that causes contraction of a muscle that removes your arm. You might need contraction of opposite muscles in order to remain balanced as you remove your foot from a tack. For example, if you step on a tack on the floor, your right leg comes up, but your left leg has to stiffen in order to maintain your balance. So you want the information going to multiple areas, but each area has its own serial processing. All right, so there is some anatomical organization in the nervous system, and we'll kind of see this as we learn different parts anatomically. I'll talk here first at the peripheral nervous system. So axons very rarely travel alone. Axons are typically in some kind of a bundle, and actually we call that bundle a nerve. That's where the term nerve comes from. Nerves are really bundles of axons that are grouped together because they go to a similar area of the body. And you can have two types of nerves in the peripheral nervous system. You can have spinal nerves. So these are nerves that are going to come off the spinal cord. These are the ones that go into your arms and legs, your chest, your back, for example. Or you can have cranial nerves. These are nerves that come off the brain itself. And these are the ones, for example, that control a lot of the muscles of the face, the muscles of the eye that we learned, sensation of the face, things like that. These are cranial nerves. And then we also give names to, so that would be white matter, right? The nerves. But we also give names to some of the gray matter in the brain, and some cell bodies like to kind of group together if they have a similar function within the brain typically, or even in the spinal cord. In the peripheral nervous, and we'll talk about that here in a minute, but in the peripheral nervous system, we also get groupings of neurons, neuron cell bodies, and they tend to occur just outside the spinal cord. And those are called ganglia, and we'll see them when we look at the anatomy of the spinal cord. We'll see some ganglia. Ganglia is the plural, ganglion is the singular. Okay, in the central nervous system, this is kind of what I was getting at, we have some areas of gray matter that like to anatomically be near each other because functionally they perform the same function. And we'll see when we cut through the brain and as we look at pictures of the brain, you can visibly see these groupings of cell bodies. They look different from their neighboring gray matter. And we call them, instead of calling them ganglia, which is what we call groups of gray matter in the peripheral nervous system, we call them nuclei when they occur in the central nervous system. Or a nucleus would be singular. And just as we had white matter bundles and axons in the peripheral nervous system, white matter bundles in the central nervous system we call tracts, and large tracts we call columns. So we're talking about the same idea, we just give them different names if they're in the peripheral nervous system versus the central nervous system. So just to give you an example of what I'm talking about, in the central nervous system, you can see here, let's look up here at this guy's brain. You can see the gray matter. So let's look kind of deeper. Let's look at this gray matter, for example, or this gray matter. We call them nuclei, right, if they're in the brain. The white matter is all where all the axons are going. So you might get a signal from the cortex that's going to go down an axon, and the axon's going to then go down the spinal cord. Remember, spinal cord is still central nervous system. So we're going to call it a tract or a column as those bundles of axons move down through the central nervous system. But once they leave, and then you might have some more cell bodies here where they synapse. As they leave the central nervous system, though, and go to the peripheral, we now have different names for them. So you may have a bundle here of gray matter in the peripheral nervous system, and we're going to call that a ganglia, right? But then out of that ganglia, you're going to have bundles of bundles of axons that leave that ganglia, and we're going to call those nerves once they leave the peripheral ganglia. All right, so this is kind of an introduction. For the rest of the nervous system, we'll be looking at the brain, the spinal cord, and learning the names of peripheral nerves as well as some of their functions as we go along. So this is kind of an introduction, and from here on out, we'll start looking at specific areas of the brain and spinal cord. I think when we meet next, I'll go over the brain and its parts in class, and then we will dissect a sheep brain. And so that should be interesting. We'll do that when we meet together for lab.